so previous to this, I talked about, I just gave a super brief overview of amino acids and as they are used to make protein. And we're going to explore that in more depth in a bit. And carbohydrates. I just very brief. I introduced them as one of the four categories in biology. The third category here is a category called lipids, L-I-P. And within lipids are fats of different sorts, fatty acids, triglycerides, all of these words that you've probably heard before. I'm going to a little bit of detail, not too much. So the fundamental building block of a lipid is a fatty acid. And a fatty acid, what would you, if you're breaking that word down, what would you guess about it? Not even looking at the picture, the structure, thinking about that. Like, if I were to say fatty acid, what would you think? Yes. Yeah. So I said, if I were to say, there's this type of compound called a fatty acid, what could you guess about? What it is? Tell me something about it, knowing what you know now. It's acidic, okay? And very simple. It was not like a complicated question. And what does that mean? I gave you guys a definition of acid. And what was that definition? Remember? Okay, pH. What is pH? What is an acid? Remember what an acid is? This is a good thing to commit to memory because it is what we have been, some of the stuff that we've been talking about. So asking a basic question, like what is an acid? Is a proton donor. So I'll write it down again. An acid is a proton donor. And a base, what is a base? Proton acceptor, yep. So in a fatty acid, there's something about it. There's going to be a chemical functionality in it that is that acts as a proton donor. And that's the acid part of it. There's also this fatty part of it, and we'll see what that means right now. So if we're going to break the molecule down, there's really two parts. There's this fatty part, and there's this acidic part. All right. So you can imagine a long carbon chain, just like carbon to carbon to carbon to carbon. Long carbon chain. In this case, the carbon chain is 1, 11, 12 carbons long, so 2, 4, 6, 8, 10, 12 carbons long, and it's what's called fully saturated, meaning it is saturated or completely full of hydrogens. All the other bonds are hydrogens. I'm not going to draw them all in. Whatever. But that last carbon is what's called an organic acid. And I've talked about those in here before. I talked about carboxylic acids before a little bit. This structure, a C double bond O, a carbonyl group, this is a carbonyl, but when it's also attached to a hydroxyl group, this whole thing becomes a carboxylic acid. And that's because a carboxylic acid can donate that proton. It can get rid of that proton, and it will look something like this. All right, where the electrons that were in this bond between the oxygen and the hydrogen come on over all the way to that oxygen and form the carboxylate group, this anionic group where the oxygen is negative and there's just a proton donated. Yeah. A double line is a double bond, two pairs of electrons being shared between the carbon, and the one line is a single pair. Yeah, I'm not going to go back and review all that stuff that we did, but yeah, this is a double bond, and this is a single. All right. So a fatty acid is capable of ionizing. This would be the acid right here, and this is what's called a conjugate base. This thing right here is a conjugate base. Because what did I say a base was? It's a proton acceptor. This carboxylate group is happy to take a proton. So protons can come on and off of it, all right, in this equilibrium between the acid and the conjugate base, between the fatty acid and the carboxylate, the fatty carboxylate, all right? This is important. It's important because this carboxylate group here is what's going to enable us to build bigger structure. Right? This ion, this conjugate base ion, is going to enable us to build bigger structure. So if we get 3 of these fatty acids, and this chain here, this fatty chain, this long hydrocarbon chain, doesn't have to be 12 long. It can be 18 long. It can be 6 long. It's usually an even number of carbons for metabolic pathway. But it's some length of carbon long, and it doesn't matter exactly how long it is. How long it is gives that fatty acid the specific characteristics that that are associated with it. So, for example, when we talk about fats, there can be saturated fats, which are animal fats, where all of the carbons are fully loaded with hydrogen. There can be unsaturated fats, where some of the carbons have double bonds in them. Those are vegetable fats. Polyunsaturated fats are like the good fats for you, where they have multiple double bonds along that backbone. They can be different chain length. This is, for example, the difference between olive oil and coconut butter, is the chain length and the degree of saturation of the fatty acid. All right? So these fatty acids, you take 3 of these things, so that's why it's times 3 here, and you combine it with this molecule called glycerol. Glycerol. Glycerol is found in it's in a lot of moisturizers because it has these 3 hydroxyl groups, these 3 alcohol groups on them, or glycerin, another name it goes by. So the fatty acid, 3 of them combines with 1 glycerol molecule, and these carboxylate groups, this conjugate base, is able to displace so the carboxylic acid, this hydrogen from the carboxylate group, is able to tear off this hydroxyl group from glycerol and form this ester. This structure here is called an ester. An ester. And then the byproduct is water. 3 molecules of water. So you can imagine taking this hydrogen and this OH, making water out of it, getting rid of it, and then this oxygen will directly connect to that glycerol backbone. And this is what we call now a triglyceride, because you have glycerol that's formed these glyceride ester bonds to the 3 different fatty acids. And each of these, A, B, and C, these different fatty chains, they can be different. They can be different. There is a wide variety of triglycerides that are possible depending upon the types of fatty acids that they're made of. All right. So triglycerides. When you get your blood tests done as you get older and you start doing like preventative medicine, you go for your checkup, they draw some blood, and they look at the blood lipids to like determine how healthy you are. One of the things they look at is what's your triglyceride level? What kinds of triglycerides? That sort of thing. Okay. This is what they're talking about. These triglycerides. So triglycerides are how fats are stored in your cell. Fat cells, also called adipocytes. Adipose tissue is tissue that is fat storage tissue. These triglycerides get packed up into there. So anyways, there's a lot more I could say about that, about fats and oils, but that's what I'm. Are there questions? Yeah. All right. So if I were to ask you on a test and say, what are the fundamentals of a triglyceride? That's like. Yeah. Glycerol and what else? Okay. Specifically, how many? 3. Yeah. So a triglyceride, the constituents of kind of takeaway that you would take from a slide like this I was talking about. What other kind of questions would I ask about this? What happened? Describe for me chemically what a fatty acid looks like. What would you say to that? Describe it. Yep. And yeah, can't stop there because that's just a hydrocarbon chain. And yeah, you can either call it an organic acid, meaning a carbon-based acid, or you can be more specific, carboxylic acid. All right, so that's how you would describe a fatty. It's a long aliphatic carbon carboxylic acid on. All right. If I were to ask you to give me a definition of an acid, what would you say to that? Someone else. Yeah. A proton donor. Great. And what's a base? That's it. You guys got it. Okay. All right. So fatty acids. Let's keep going. So triglycerides themselves are just building blocks for bigger things. All right. So we can make these things called phosphoglycerides out of them. These are very important. These are the lipids that make up the membranes of your cells, the outer and inner membrane, the plasma membrane of your cell, these phosphoglycerides. Phosphoglycerides themselves can be derivatized into bigger structures, like glycerosphingolipid, whatever. There's all these other things. But the way you go from a triglyceride to a phosphoglyceride is you take a triglyceride and you add a phosphate group. A phosphate group is a phosphorus-based anion, meaning a negatively charged ion. And the phosphate group displaces one of these fatty acids, basically kicks this bad boy off, and comes in. So phosphate looks something like this. Like that. That's what a phosphate group looks like. And this oxygen would be this oxygen. So in a phospholipid, it would look like this, CH2, C, and CH, and then this would be the other CH2. H2. So that would be a phospholipid. It's important here. What did we do? What did we do to the structure? How would you describe the difference between the structure of a triglyceride and a phosphoglyceride? Using like your best chemical language that you describe that. Using the terminology of polar and non-polar. Yeah. What were you going to say? Great, absolutely. And how? What sort of change does that phosphate group bring? Would you guess? Yeah. 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 So that's right. They are opposites, and the word for that is amphipathic. Amphipathic is the word for that. But this polar head group. What does polar mean even? Polar does not mean they're opposite, but in terms of what? What does it mean polar? Is polar something that's polarized? Does that mean that they believe in it or don't? What does polarized mean? Okay. I don't know what it is. Okay. Yeah. Anybody else want to take a stab at it? Stick their toe in the water. Okay. Something that's polar is or polarizable is something where, in the most simple term, it's something that likes water. All right. It's something that will go into solution in water. If it's polarizable, it's something that likes to be in. And a little bit more sophisticated description. Imagine you have something right here, and something that's polarizable. You can have, like, what does it mean? I guess that the country is polarized when we use that word to describe people. What does that mean? What's that? They're separated by what? Whatever, yeah, whatever their differences are. So maybe we have all the people that believe that summer is best over there, and all the people that believe really winter is where it's at on this side. And the class has been polarized, and we like, you are crazy. Winter is the awesomest. No, summer. It's so nice to not have to wear a ton of clothes. And so people separate along those lines, right? A molecule that's polarizable, it's polarized along electron density. All right, so there's a lot of electron density maybe here and not much here. So there's a little bit. It's like becomes polar. Yeah. Yep. The dipole moment of water. Right? There's a dipole moment to water. Water itself is polar. And because of that, it likes to have polar things in solution. The ability to like, yeah, the ability to have a polarizable electron cloud. So, for example, here in this carbon chain, carbon and hydrogen. Let me show you this. If we look at the periodic table, this is the color in this periodic table represents how electronegative, how greedy an atom is for electrons, how much it wants to rip the electrons to it. And it turns out that, and they have these numbers associated with them. You can't see them very well. Maybe I can make it bigger. I do. View image. Okay. So if you look at this, they have these numbers here. You can actually quantify electronegativity. And you can see that fluorine is the most electronegative. So hydrofluoric acid is very strong acid because that's very polar. Carbon and hydrogen are similar electronegativities. So a greasy tail that has carbon with hydrogen on it. Hey, what's up? Hey, okay. A carbon-hydrogen bond is not very polarized because in that tug of war of electrons, they're about as strong as one another. It's not very polarizable. Whereas an oxygen-hydrogen bond, oxygen has a 3.5 electronegativity versus a 2.2 for hydrogen. So oxygen wins. Electron density goes this way, and we get a negative and a positive. All right. And water, because it's just hydrogen and oxygen, is very polarizable. It's a polar solvent. Am I blowing your minds? This is, again, just some chemistry. Yeah. Yeah, this is a little bit far afield, but those are noble gases. And because they are noble gases, they don't really, they satisfy the octet rule. They have a complete electron. Their valence shell is completely filled. They don't bond. They're not really considered. Yeah. For purposes of this, yes, I'll give you a definition of polar. So a broader definition of something that is polar or polarizable is that it is subject to a distortion of an atom or a molecule whose electron cloud is not even spatially. Does that make sense? That's like the more explicit. For this class, what that means is that things that are polar are going to be things that like to be in water. Things that are non-polar are going to be things that want to get away from water and do not like to be around. Does that make sense? Okay. So I mean, imagine it's like this. That's exactly why oil. The fatty acids and the triglycerides. What's that? Oil shouldn't have much of any water. There's very, very little water. Okay. So the thing about a phospholipid is that you've taken this pretty nonpolar molecule. I mean, there's some oxygens in it. It's not completely nonpolar. Triglycerides, for all intents and purposes, mostly nonpolar because you got these long greasy tails, this sort of like greasy carbon chain here. We've got a couple, ester groups in there, but ester groups are not super polar themselves. So we just rip one of these things off. We stick a superpolar phosphate group on there through what's called a phosphoester bond. And now we have something that's very polar on one end and non-polar on the other end. And they're able to line themselves up in such a way that they form these lipid bilayers. This is how we build cells. All right? This is how cells get built. So we have all of these negative charges, these like very polar head groups along one side, and then they hide all the greasy tails on the inside of the lipid bilayer. So there's an inside and an outside membrane. You can imagine this extending out and going into a big circle, which is the shape of it. Does that make sense? I mean, you should have talked about that in biology. Well, welcome to biology. That's phosphoglycerides are why we one of them. Okay. And there's obviously more that we can do there, but I'm giving you just the very basic building of life. These biomolecules. Any questions on this? So let me ask you some questions so you can understand. Because one of the things that I want is for you to be able to look at my slides and be able to say, oh, this is what's important. This is what I need to know, because I could expect these kinds of questions on the slide. Right? I'm not trying to fool you. What I'm trying to give you information. The only reason I test you is to see if you are hearing what I'm giving you. So the kind of question I would ask over a slide like this would be something like, well, just like some basic stuff like, what are the components of a phosphoglyceride? And you would say a triglyceride, one triglyceride, and a phosphate. That would be the super basic. That's just sort of regurgitated. And then I would ask maybe a higher order question. So, for example, describe for me a property of a sort of biophysical property of a phosphoglyceride? And you would say, well, it's polar on one end near the phosphate group and non-polar on the other end, which enables it to form these lipid bilayers. Yeah. Phosphoglycerides always have the phosphate group attached. You can take phosphoglycerides and make other things out of them, which is, maybe we'll get to that, maybe we won't. I don't know how much biochemistry. I'm just trying to give you some. All right. So now I wanted to start talking about going into a little bit more depth about protein. Because to understand what's happening in your body, you have to understand the basics of an enzyme. And I often start with the nitrogen cycle review, which I don't know if it's a review or not, but that's what you have in a bio. The basic idea, I'm not going to go into this in detail, but the basic idea here is that so amino acids, you remember amino acids have the amino group, the carboxylic acid group, the alpha carbon that's in the middle that has its function. That nitrogen, that amino group has nitrogen in it, and that nitrogen, all the protein in your body, in every organism that's alive on Earth, it comes from nitrogen gas that's in the atmosphere. 70% of the air you breathe is nitrogen. And that nitrogen gas is not actually available to most organisms. So we have to get it into a form that can be assimilated into an organism. That's called nitrogen fixation. So there's this cycle that happens where there's nitrogen gas, nitrogen fixation, oxidation, and then denitrification. It goes around and around. This ammonia, though, is able to be taken up by different plants and bacteria that are in the soil. That ammonia is then used by plants and bacteria to make amino acids, which get incorporated into protein. building things. So in general, these two words are good to know. We'll use them again later. Anabolism and catabolism. You guys have probably heard of anabolic steroid before. Anabolism is the process of building something bigger up from something smaller. So taking small Legos and building a tower. Anabolic process. It's anabolism. And taking big things and breaking them down into their smaller constituent parts is catabolism. So this back and forth is anabolism and catabolism. Those are the two different arms of metabolism. All right, metabolism is made up is the broad. A person's metabolism is made of anabolic and catabolic processes. So metabolic processes are either anabolic or catabolic. Does that make sense? Metabolic. Okay, so that's the nitrogen cycle review. The takeaway from this is, so if in your note-taking, remember at the beginning of the year I talked about ways of taking notes, write all your stuff down, go back and processing it and like, oh, what was the point that Dr. was making? The takeaway here is that the nitrogen that we use to make amino acids and then ultimately proteins comes from nitrogen atmosphere that is fixed by plants and bacteria. All right. So again, to remind you, here's the structure of an amino acid. We've already kind of gone through this, but I guess to review it, here's the amine group, here's the carboxylic acid group, and here's the alpha carbon, which has the side chain. This side chain R can be 20 different side chains that make up the amino acid of a person. All right, amino acid. It's an amino group, it has an acid group. So the kind of question I would ask about a slide like this is, what are the chemical constituent parts of amino acid? You would say, well, an amino acid has an amine group, it has an acid group, specifically a carboxylic acid group, and it has another carbon, an alpha carbon, that has a side chain. Parts. Yeah, they call it alpha, an alpha carbon, because it's alpha I guess nitrogen is where you start the counting. So it's alpha. It's alpha. So like in a carbon chain, for example, in like a fatty acid, they're going to count the carbons in that chain, and they count it using the Greek alphabet. The alpha, the beta, the gamma, the epsilon, etc. You've heard of like omega 3. So omega would be the last carbon, the last letter. 3 is 3 off of the omega carbon, or that site of the double bond. And in an amino acid, there only is an alpha carbon. There's not other carbons in this chain. Here's carboxylic acid. Here's the alpha carbon. It just refers to the carbon that's not the carboxylic acid. Alpha. Good questions. All right. So all this talk about anions and acid proton donor is important because this is how we build proteins in the cell. Here's an amino acid of some sort. I'm going to put a R group here. There's no R group there written in, but whatever it is, it could be any kind of structure. Doesn't matter. One of the 20. Here's the unionized amino acid. And then it's able to form what's called a Zwitter ion. So who speaks German? Anyone? Count in German. Who can count in German? 1, 2, 3, 4, 5, 6, 7. That's German. Yeah. Well, it was Germans that came up with this word. Anyways, Eins, zwei. Zwei means two. So Zweiter ion is the ion that's got two different charges. Eins, zwei. Zwitter ion. So the Zwitter ionic form. Yeah, totally weird diphthong there. ZW. ZW. But there's one.
So
it's got both the amino group ionized, where we have added another hydrogen to it because nitrogen doesn't mind being positively charged. You add a proton to it. So over here, the valence shell in nitrogen. Nitrogen is atomic number 7 for nitrogen, right? And that means the first shell has got 2 electrons in it. Second shell has 1, 2, 3, 4, 5, and so in the valence shell, there's going to be 1, 2, 3, 4, 5 electrons in that nitrogen. So in an uncharged nitrogen, and here's a hydrogen, here's a hydrogen. The other electrons come from the hydrogens and the carbon. There's a lone pair of electrons right there. But the nitrogen doesn't mind unloading those a little bit because it's got not the super strongest electronegativity. And this electron can get picked up by a hydrogen ion. And so it can form this. You can protonate. It acts at, would this amino group be, would you consider it to be an acid or a base? Using the definition, here's a good question. Would you consider the amino group to be an acid or a base, following our definition of acid and base?